Christian, thank you very much for the opportunity, and thank you all for attending this, if I believe right, the last or the final talk, the last or the first talk, right, depending on what event you are attending right now. And indeed, I'm speaking from Budapest now, where, as Christian said, I'm splitting my time. Now it's starting the Budapest time for the next few months. And you know, the question I would like to address today is a little bit kind of close to our heart, which is what exactly did you do during COVID? And today's talk really would describe the process of what we have actually gone through during the COVID, what my lab has been busy with, and including the timeline. And hopefully I will convince you that it was time well spent. Now, I start the title with network medicine. And so let's first say a few words about what network medicine is, why do we need a new word, and what we really kind of try to, what tools that we try to use there. And kind of thinking about network medicine, at least in my lab, it started around 2007 or so when I was first at Harvard at the Dana-Farber Cancer Institute on a sabbatical. And it was the time soon after the genome project was finished, a few years after, and it was a time where every week another gene was connected to another disease. The so-called disease genes were just discovered. And one of the questions we asked at that moment is that, how do we really conceptualize these disease-gene relationships? The fact that, that there are cancer genes and there are deafness genes and there are diabetes genes and so on. And we came up with this particular representation, which now we call simply a bipartite network representation, where we simply connected diseases if the same gene was responsible for the emergence of both. That is, if the mutations in gene A could cause both, say, deafness or cardiomyopathy, then you could actually have a link between them. Why was this link meaningful? Because this link was actually indicating that the two diseases have common genetic origins. And so we ended up with this very interesting map where the node sizes correspond to different diseases, and the size represents how many genes were at that moment associated with that particular disease. The colors correspond to the class of diseases. So for example, here on the left-hand side, you're seeing lots of blue-green nodes, and those are all cancers. And you see that there are lots of links between the cancers because there are quite a number of genes that are involved in multiple cancers, like the famous P54 gene that is typically implicated about almost 50% of the cancer occurrences. And so we actually had lots of fun with this map, but this is what I would call the network medicine 1.0, because it looks like a 20,000-mile view of how diseases are connected to genes. Soon after, we started what I would call Diseasome 2.0, which is to find out where do these connections come from. And what you see in this video, and hopefully plays well on your computer as well, is what we call the subcellular network, or the technical term is often the interactome. And the interactome is nothing but all the physical interactions between the proteins within our cells. As you know, the proteins are not inert molecules within our cells, but they bind to each other, and that binding leads to molecular machineries, and those molecular machineries are responsible for many functions in the cell. And the breakdown of these molecular machineries is often responsible for what we perceive as disease. So Diseasome 2.0, or network medicine in general, is trying to understand how these connectedness within the cell and the breakdown of the connectedness within our cell leads to identifiable diseases. And how do we really kind of connect this network to the particular diseases that we may get? So this network is very large. It has about 20,000 proteins, about 20,000 genes in them, and hundreds of thousands of experimentally identified physical interactions or binding interactions between pairs of proteins. Now, the question is, what does it mean to have a disease? Where is a disease localized within this particular network? Well, we have the concept of disease genes, and we can therefore start talking about where is a particular disease located. Now, to understand the concept, I'm going to switch away from this particular network, and I'm going to let you to suspend disbelief and come with me and believe that the network that I'm going to show you is nothing but the map of the cell. And that's the network. And of course, you all recognize this as being the grid of New York City, the map of New York City. And let's for a moment believe that each intersection of the roads corresponds to a protein within our cell, and the road segments would correspond to protein-protein interactions, just to fix certain ideas and how we think about disease. Now, if we do that, the question is, how is function distributed within this network? And if you've been to New York City, and I'm sure many of you have been, and in case you were interested, and many tourists do, were to go to see a Broadway play, what you realize that you don't go just anywhere within this network to watch a Broadway play, but there's a well-defined neighborhood where all the theaters are located. And even though Broadway is a road that really crosses across all Manhattan from south to north, right? The Broadway that we perceive as the theater or the theater district is well localized. So you would say that the function of theater within Manhattan is well localized in a certain network neighborhood. It turns out it's not only theater. If you're an art lover like myself, you would actually realize that most of the high-end galleries are located in a different neighborhood of the map, which is what we call the meatpacking district traditionally. It's kind of on the left here. And while there are a few galleries scattered around the city, really, if you are interested in art, you will start in that particular network neighborhood. So what this map suggests is that in Manhattan, just like in the real cell, functions are not randomly distributed throughout the network, but they are well-defined network neighborhoods. You can call them disease or functional modules that carry particular functions. So now, if we believe the functional module concept, then we need to ask, what does it mean to have a disease? And typically, a disease is nothing but the breakdown of a certain function within the cell. So this would suggest that the disease genes would not be localized just about anywhere in the network, but disease genes associated with a particular disease are in the same network neighborhood. So carrying for a moment again the Manhattan analogy, we would say that cancer is located in the vicinity of Wall Street. And we may put bipolar disorder around Times Square. And maybe let's put asthma, another disease, around Washington Bridge, where Manhattan breeds much of its mass into New Jersey. So the idea is that then the disease genes correspond to the breakdown of a function. And because function is well localized, so will the disease modules be well localized. Now, if you believe in this, then if you want to understand the disease, then the situation is very simple. You need to get a map of the cellular network, find within that where are the disease modules. And once you identify the disease modules, then you need to perturb it or drug it. That is, find a drug that hits in the right neighborhood of the disease module that can perturb the function and perhaps alleviate the symptoms of disease or restore functionality. So that's kind of the elevator pitch, the big picture. But let's go back to the cell and ask ourselves, is it really true that the disease genes are well agglomerated in a certain network neighborhood? And to illustrate that, what I'm showing you is the map that I showed you earlier of all the protein-protein interactions in the cell. And now I highlight in purple the disease genes associated with it, asthma, one kind of a respiratory disease that we study a lot in my lab. And what you see is that many of these asthma genes are connected to each other and they reside in the same network neighborhood. And a few of them are disconnected, and sometimes they're disconnected because we're lacking some protein interactions that may connect them. Some of these networks are inherently incomplete. There are many interactions that have not been experimentally discovered. And sometimes some of the asthma genes are disconnected because they may not actually be asthma genes. So some experiments suggested that they are associated with asthma, but maybe that experiment was faulty or the association wasn't strong enough to be really validated. But the bottom line is that we can actually see that there's a particular network neighborhood where asthma resides. And that's what we call the disease module or the asthma disease module. Now we have this conceptual framework that each disease has a well-defined neighborhood in this network. Let me go to the topic of my talk, which is actually COVID-19. Now, COVID-19 is a complicated situation because it's not something that inherently happens within our cells, but it is caused by a pathogen, which is a virus that interacts with our cells. So the question is, how do we conceptualize the COVID-19 disease as from a network perspective? And this is something that we've been working about 5-6 years ago a lot in my lab. We had a center, a genomics center that was actually focusing on the very question of virus-host interactions, host being the human cell, virus obviously is the virus. So what is a virus? Well, a virus actually consists of several proteins, and I'm showing here the virus here. And in the case of the COVID-19, there are about 29 different proteins that are coming with the virus. And these proteins get injected into the cell. And this is the human interactome. This is the network that I showed you earlier. And when they get injected, some of the viral proteins bind to human proteins. So they have protein, virus-host protein interaction. So this is the binding interaction. And by binding, they induce perturbations in the human cell. And those perturbations, those bindings can cause loss of certain function in the cell. For example, the virus proteins can turn off certain processes that would otherwise kill the virus and can accelerate other processes, like the production of certain metabolites and proteins that the virus needs. So the way we perceive the virus-host interaction is that we have now kind of several 2,000 new proteins that enter the cell, and some of these viral proteins interact with some of the human proteins, and most other human proteins are not touched by the virus whatsoever. So this leads to the concept of the COVID-19 disease module, which is the network neighborhood where the virus hits. And the question is there such a well-defined network neighborhood, or the virus hits randomly anywhere in the network? So that's the first question we need to answer. So let's look at the timeline for our COVID-19 work. So what happened in our case is that the shutdown happened around March 15 or so, but it was on March 23rd when the first piece of data became available to us experimentally, which is what are the proteins that the COVID-19 or the SARS-CoV protein attacks? What are the human proteins that the virus interacts with? So that became available on March 23, and that's when we started our project because we had the starting data. And our starting project involved the map that we've been curating for many years, which is the human interactome. This is all the protein-protein interactions in the human cell, interactions between 18,000 proteins, altogether 332,000 links. And then the SARS-CoV-2 targets, and these are 320 human proteins. So the 29 proteins that are in the SARS-CoV virus interact all together with 320 human proteins. And then, of course, the question we wanted to ask is that what are the drugs that are available on the market and are clinically approved that could actually perturb the virus's ability to infect the human cell? So our starting point was data set from the drug bank of 7,500 drugs, altogether having 4,100 drug targets. The drug targets are human proteins to which the specific drug binds to. That can be one or multiple. And you may ask, why are there fewer drug targets than how many drugs are there? That is because many, many drugs target the same target. That is, somebody discovers that a certain protein is a good drug target, then many companies come up with different drugs to target the same protein. So that was the first step. And then the first question I'm going to ask is that, do we have such a thing as the COVID-19 disease module? And for that, we took the 322 human proteins to which the 26 SARS-CoV proteins bind. And then we ask, how are they distributed within the network? And what we learned is that 208 of these 222 form a large connected component, that they are connected to each other in a single connected subgraph. And this large connected component could not emerge by chance because it has a z-score for 1.65, so it's outside of the sigma region. So indicating that really there is a well-defined network neighborhood where the SARS-CoV-2 virus attacks. So their targets is not random within the network, but well localized, just like the asthma genes were well localized in the large network. Now, we did that for the full interactome. We could also ask what happens if we only look at lung. Now, what's the difference between the full network and lung? Well, in the lung cells, not all proteins are present. It's called actually that not all proteins are being expressed. So if you want to study only lung, where typically the SARS virus attacks, you need to restrict the network to the subset of nodes that are present or expressed in the lung. And that's what we did over here. And so some of the nodes you may see are kind of like shown in yellow because they are not expressed in the lung. Those who are expressed are shown in the green. And you still see that you have a large connected component. And it indicates that in the lung, there is still a statistically significant disease module. Why is that important? It's important because a few years ago, in this paper, Kitsak et al., we have actually shown that typically a disease manifests itself in the tissue where there is a statistically significant disease module. That is, this result explains that why don't you, for example, have asthma in your brain? Why do you have it only in your lungs? Because in your lung, the asthma disease module is there, but in your brain, even though many of the asthma genes are expressed, they don't form a disease module. So this methodology allows us to ask, what tissues could the virus infect? What are the tissues for which the virus have the right interaction partners? And when we did that, we actually learned that lung is among those. But interestingly, we also found that the disease module necessarily for the virus to attack is very strongly present in brain, as well as in many cardiovascular tissues. And this was actually, I remember we kind of released this one like late March, early April. At that time, there was not much experimental clinical evidence that the virus could attack the brain. In the meantime, we have lots of evidence that indeed the COVID virus could penetrate the brain and could actually cause differences there as well. And we also predicted that cardiovascular tissue should be affected. And we know that many of the comorbidity of the COVID virus is cardiovascular. So that's kind of one aspect of it. What could you use the network to tell you what tissues could the virus affect? But we can also ask questions. What other diseases are in the network neighborhood of a particular disease or where the virus attacks? And here I'm illustrating, I go back to asthma to illustrate this. So on purple, I'm showing again the asthma genes. And in yellow, I'm showing the so-called COPD genes. COPD is another lung disease. It's often called the smoker's disease because among those who have the disease, there's a very, very high percentage of smokers. You can be non-smoker and get it, but typically smokers get that. And he has very similar symptoms as asthma, but it has different clinical manifestations, that the way the lung breaks down is very different. And that being said, what we find is that the COPD genes are in the network neighborhood of the asthma genes. And to us, this actually suggests that diseases who are similar to each other happen to be sitting in the same network neighborhood. And we tested that in this paper by Mencha et al., my former postdoc, who is now a professor in Vienna, by developing actually a measure to determine what is the distance between two diseases in the network. And in this way, we were able to effectively place all diseases in this kind of network and asking the question, are the two diseases overlapping or not? So here, each sphere corresponds to a disease. The size of the disease corresponds to the size of the disease module. And two circles or two spheres are overlapping if their disease modules overlap. And what our previous results from this 2015 science paper indicates is that the more two disease spheres overlap, the more two disease modules overlap, the higher the so-called comorbidity between them. That is, if you have one, you're likely to get the other disease, as well as they have many common symptoms and so on. So therefore, we could ask the question, is there a chance that there's a particular disease module that the COVID attacks? That could explain the symptoms it causes? And the answer is no. That is what we learned by measuring the distance between the COVID disease module and all other diseases is that the COVID doesn't attack directly any existing disease module. But it has a couple of disease modules that are close to it. And some of them are known to actually be kind of showing up if somebody's infected with the COVID, like cardiovascular diseases and so on. But there is no particular disease module that is being attacked. Why is that important? If the COVID would attack a particular disease module, A, it would have the symptoms of the disease, and B, the drugs available to that disease would be very good candidates to cure the disease. But because it doesn't attack any module, we need to develop other methodologies that don't just rely on a single disease, that is disease-blind methodologies to identify what drugs could be effective against COVID-19. So how do we do that? And to understand how we do that, we're going to get to the step 2. So after we identified that there is a disease module, then we ended up implementing 3 classes of what we call drug repurposing methodologies. And drug repurposing methodologies are methodologies that rely on the underlying network and connectedness of the genes within the cell, and try to look at what are the drugs and where the drugs hit the network, what are the nodes that the drugs perturb, and try to predict what of the existing drugs that are already approved for human use could be actually affecting the disease that is caused by COVID-19. And typically the underlying hypothesis is that we're looking for drugs that hit in the vicinity of the COVID-19 disease module, that is hit in the same network neighborhood where the virus perturbs the cell. And by doing so, it may perturb the virus's ability to infect the cell or to replicate within the infected cell. So what are the 3 methodologies that we used? One of them is what we call a proximity-based ranking, which simply is shown over here. If these are some of the genes that the virus perturbs, so this is the disease module associated with the virus, and these are the drug targets. These are the proteins that the known drugs are targeting. We would like to measure the distance between the drug target and what we call the disease genes, in this case, that are the genes associated that are being targeted by the virus. And what we have shown many years ago in 2016 in this Gunri et al. Nature Communications paper is that there's a particular measure which relies effectively on the statistical significance of the distance. That is, how far are these distances compared to a random selection of nodes, could actually predict whether a drug hits in the right network neighborhood or not. So effectively, we measure the distances between the drug target and the SARS targets. And then we say, are these distances shorter or larger than what you would expect if these drugs would hit randomly on the network? And that gives us a Z-score that gives us a statistical significance of how close the particular disease is hitting to the disease network. So that's methodology one, and we're going to call it the proximity-based methodology, or P. We also implemented a diffusion model, which effectively tries to identify nodes whose relationship to the disease genes is kind of very similar to other cases where a drug was effective against a certain disease. And to do so, it effectively implements a diffusion-based model on the network, and that methodology is kind of telling us whether the drug is hitting in the right way or not, in the right neighborhood or not. And the third methodology came from Marinka Zitnik, who has published in the last few years a series of papers where she used graph neural networks to predict drug repurposing opportunities, that predict drugs that work for other diseases. And effectively, this creates an embedding in a high-dimensional state between drugs and the disease genes. And when you are faced with a new drug-disease combination, the neural network is trying to kind of decide whether the patterns that is seen in previous drug-disease relationship that worked is similar to the one that you are testing right now. So we have 3 different methodologies, and we're going to call it the AI methodology. This is the last that I showed you, the diffusion and the proximity, or A, D, and P. And you can implement these methodologies with different parameter sets. So at the end, the AI has actually provided 4 ranking lists, because each methodology provides not simply a yes or no answer, but it offers what we call a ranking list. It takes all the drugs that it can predict on, and then it ranks them that this is the most likely to be effective, this is the second most likely, and so on. And the AI has predicted ranking for 1,670 drugs out of the 7,000 that we had in the drug bank. The diffusion has offered predictions for 6,000 drugs, but it offered 5 different rank lists based on the way the diffusion method was implemented. And the proximity methods, the first that I showed, that are simply looking at the distance between the disease genes and the SARS targets, has offered 3 different ranking lists with different number of drugs. The shortest is the P3 because P3 was different. It didn't rely on drug targets, but it relied actually on differentially expressed genes caused by the drugs. So it's a detail, but it was a little different data that went as an input. Now, so effectively each methodology has provided a different ranking list. Altogether, we have 12 different ranking lists. And these ranking lists are not independent of each other because they all act on the same input data. They all predict from the same network, from the same list of drugs with some differences, and the drugs have the same targets. And we purposefully did that because we don't know which method works the best. So we wanted to compare the outcome of each of the methods to be able to decide which one to use, which one with the most accurate in predicting what drug works for COVID. And the fact that they are not independent of each other is shown over here, which shows the correlations between the rankings provided by the disease, by each pipeline. And it shows that the diffusion rankings are highly correlated to each other. So are the AI rankings among themselves are highly correlated, that the list is very similar, not exactly the same, but very similar. And 2 of the proximity was similar. And of course, the P3, which relied on different data, provided yet another independent set. So we have altogether 4 very different set of predictions, but within a class, there is lots of similarities. So this is actually the end of the prediction pipeline. And I want to tell you where we are now in the timeline. So this work was finished between March 23 and April 15. And by April 15, we had the 5 pipelines. We had the predictions provided by them. And then the question was, how do we know who is right? And in order to know who is right, you need ground truth. That is, you need independent data to tell you which of the drugs have really efficacy among those that were predicted by the pipelines. And to achieve that ground truth, we relied on 2 different types of data sets. The first data set is, and I'm going to describe them actually. I'm not going to talk much about this third one, but these 2 I will talk quite a bit. The first one was what we call the clinical trials data, and the second one what we call experimental outcome, and I'll tell you where that comes from. And to obtain that was actually April, May 15, and June 15. It was a 3-month work, and you will see why was that. So the first was actually what it took much longer was experimental outcomes. So how did we experimentally test our predictions? Well, I showed you that the different pipelines predicted different number of drugs, but there were altogether 918 drugs for which each of the pipeline that we tested made a prediction. So, and the remaining 6,000, some of the methods made predictions for some didn't. So we selected these 918 drugs, whether the method predicting to be likely to work for SARS or whether it predicted not to work. We didn't care about the outcomes. We just cared about the fact that all methodologies provided predictions because we needed such a set of drugs to really compare them. Because if they don't predict for the set of drugs, the same set of drugs, then we have difficulty comparing the outcomes for them. And also there was a second criteria that defined the 918 sets is that the chemicals, that is the drugs themselves, were available for experimental testing at the Broad Institute at MIT. So we could actually do experiments. We mean actually the Boston University Infectious Disease Center, which is one of the few places in the world where people actually in scaphanders can work with very dangerous viruses. And you will see a picture soon of our collaborator doing that. And they have taken these 918 drugs and many others, but they're not relevant for us. And they have experimentally tested by effectively adding them to cell lines that were infected with the virus and seeing whether by adding the drug itself kills the virus or not. And these experiments were done in what we call Viro E6 cells, which are derived from monkeys, African green monkeys. And the only reason they were done in this, because this is what experimenters call a very stable system that can be easily infected by the virus. Human cells are much harder to experiment with. Simply the virus is very difficult to induce an infection. So this is what many experiments are focusing on. And what kind of experimental outcomes can we get? The best case is what we call a strong case. So what you see actually here on the horizontal axis is more and more drug being added to a cell line. That is that, you know, like you're adding more and more and increasing the efficacy potentially. And if the red line actually goes down, it means that the number of infected cells have gone down. And that's what's a good outcome. And here I'm showing you two results, which we call strong, because they have significantly reduced the count of the infected cells. And the blue line corresponds to the fact that they have not killed otherwise the uninfected cells. Some drugs actually work by simply killing all the cells, whether they're infected or not. So these are what we call the strong results. And that's what you're looking for typically, drugs that can kill the infected cells, but don't kill the normal cells. Some drugs kill some infected cells, but not enough. And this we call weak drugs. And altogether, on the 918 drugs, we found 37 to be actually strong and 40 to be weak. And weak means it had an effect, but it wasn't as impressive as we hoped or needed for experimental, for using it in the clinic. And then there were two more classes that really matter for us. One of them is the cytotoxic. These are drugs that just kill the cell, whether it's infected or not. And of course, we know that they will not kill you because these are approved drugs. But nevertheless, for these, you cannot decide whether it's effective or not against the virus, because in the experiment, it killed the cells. There are only 35 of them. We will simply ignore them. And then there were altogether 806 of the 918 drugs that had no effect whatsoever on the virus. So these are what we call no effect drugs. And to move forward, we will consider positive outcomes or ground truth, the strong and the weak drugs, because both of them have affected the virus's ability to infect our cells, weakly or strongly, and there are altogether 77 such drugs. And we will consider as negative outcomes the 806 drugs on the right-hand side, those actually who had no effect on the drug. So this was the experimental validation, and we will use this data to test how our pipelines worked. And this took about 3 months to achieve, to test this many drugs. It was a heroic effort, and we were impatiently waiting on the side, but we had to wait for it. Now, the second set was what we call clinical trial validation, which is when we finish the predictions, and this was important because we wanted to set a blind prediction situation up, where first we make the predictions and then we actually start getting the evidence so that the predictions are not affected by the available evidence. So on April 15, 2020, when the predictions were done, we scanned clinicaltrials.gov and identified all the drugs that were at that moment under clinical trial. And altogether finding about 67 drugs. And we again limit our analysis to those that were also tested experimentally. So that's just a minor detail. And what I want to emphasize that what kind of ground truth this data is. Well, we don't know whether these drugs are effective or not against the virus. But we know that the medical community thinks that these drugs should be effective against the virus, because that's why they're investing millions of dollars to do a clinical trial. So this particular clinical trial data set is really testing our ability to reproduce what the medical community believes that could be an effective drug. So these are going to be the two data sets, and the experimental outcomes and the clinical trials. And I will refer to it as E918 dataset based on the experimental and the 918 drugs. And then the CT415 dataset, which is the clinical trials collected at April 15. And the first question you want to ask in any prediction set is, what is your AUC? That is, what is the area under curve, right, the outcome, because that's a standard measure that the prediction literature used to kind of compare different methodologies. And the good news is what we see is that most methods, not all, so what you see here is the 12 pipeline. You remember the 3 AI-based pipeline, 4 AI-based pipeline, 5 diffusion-based pipelines, and the proximity, 3 proximity pipelines. And on the vertical axis, I'm showing you the AUC, that is their ability to predict, you know, the experimental outcomes. And what we see is that there are differences between them. Most pipelines are predictive. That is that the outcome, the AUC is above 0.5. The AUC numbers are not particularly impressive in the experiments, but we never expect in the biological setting to get an AUC of 0.9 or so. That is unrealistic in this context. And what we see is that some pipelines do very well for the experiments, like the P1, for example, over here, the proximity one, and so does the diffusion 2 and so on, and others do much worse, like diffusion 3 and diffusion 5. We see a very different picture for the clinical trials, where the AI pipelines do very well with an AUC of between 0.7 and 0.8. And now the diffusion pipelines do poorly, and some of the P pipelines do even worse, and some of them do all right. So you see there's no consensus. So in one data set, one methodologies work, and the other data set, some other methodologies work very well. Now, of course, the trick over here is that we're looking at the AUC, but in an experimental setting like what we have, the AUC is not really the right measure. Because the goal of drug repurposing is really to prioritize all available drugs and to tell you what are the few that you should take for further experimentation. However, the AUC is a measure that extracts its predictive power not only from the top of the list, but also from the bottom of the list. In a drug prediction settings, we don't care what's at the bottom. We just care about getting right who is at the top. That can we use the network methodologies to predict who are the best drugs. So therefore, precision at 100 and recall at 100 are much better tools to really predict, much better measures to tell us what should work and what doesn't. And as you know, precision at 100, we're asking how many of the drugs that are effective were chosen among the top 100 by the particular methodology. And that's what I'm showing you over here. And for the experiments, you see that actually the AI has done the best when it comes to precision 100 by choosing 9 effective drugs among the top 100. And some of the AIs have actually chosen as many as 6 or 7, and so did some of the proximity methods. And when it comes to the clinical trials, again, the AI does very well. The diffusion does very, very poorly, effectively no predictive power, and mixed results actually for the P methodologies. We get very, very similar results for the recall. That is how many of the total effective drugs were actually captured among the top 100. And again, seeing a very mixed outcomes. So when you put all of these pieces of results together, we actually get some very mixed results. And the mixed result is that there is not a single methodology or pipeline that is reliable across all measures and across all data sets. They all have predictive powers by themselves, and together they can actually predict lots of good outcomes, but we don't know which one to actually trust. So therefore, in the second part of the work, we actually developed a multimodal approach, and I'm mindful that I'm short in time, so I'm gonna go a little faster here. And with the multimodal approach, what we asked is that in reality, what we have is that you have experimental resources to follow up on, say, K drugs, say 100 or 120. And then what we would like to know is how do we select these top 100 drugs? What methodology do we use so that we get the best outcome? And one possibility would be that if we have n pipeline, we have 12 pipelines, you just take the top k over n fraction of each pipeline. So say, take the top 10 results from each pipeline, and for the 12 pipeline, we have 120 drugs, and check those 120 experimentally. The other possibility would be not just to take the tops, but take for each drug the average ranking across all the methods. This is what we call average ranking. And this is very widely used in the literature. Both methods are being used in the medical literature. We actually developed an alternative approach, which is based on ranking and election counting, is that how do you actually decide on a good election outcome? And we're relying on what is known in the literature as the Kemming consensus. That is, we don't really care who's number one or number two in the particular pipeline, but we care what are the pairwise agreements on each pipeline. That pipeline one ranks A better than B, and if pipeline two also ranks A better than B, doesn't matter where they are on the list, as we use this relative ranking to each other to come up with an outcome. This is known as the Kemming consensus, the optimal outcome, which is finding the list that finds the best agreement across all the pipelines. The problem with the Kemming consensus is that it is NP hard to complete. So, and given the size of our ranking list, it's impossible to calculate all the possible versions. So we actually going to rely on a couple of rank aggregation algorithms that try to approximate the Kemming consensus, so-called Bordas count, Downwall method, and C-rank, a Bayesian method. So the question we ask, if we can test only 120 drugs, what would be the best methodology? The union list, the average rank list, or one of the consensus pipelines? And the outcome is shown over here. So this is horizontally how many drugs we have to test, and vertically what's the recall of the particular list. And to guide you through, what we find is the average rank, the lower it is, the worse the results. And what we find that the average rank gives you the worst results. So if you just simply calculate the rank of the drugs across all the pipelines and you average that, that's the worst outcome. The union and Downwall do a little better. The union is when you just take the top of every single list. But really the Borda count and the C-rank does the best. And generally, whether we look at recall or whether we look at the experimental data or whether we look at the clinical trial data, we find that the C-rank, which is the Bayesian method, consistently beats all the methods. And what you see that C-rank is able to combine all of these different independent set of predictions and come up with an outcome that beats every single individual pipeline and gets the best predictions from all of them. So it a little bit works like magic. And this was really the methodology that we relied on to really kind of come up with the final ranking list of the drugs. So this is kind of one of our key results, is a methodology of how you can combine by seeking a Kemming consensus, the different independent predictions. And then the predictive power from the combination is better than any individual pipeline. And I think this is very important for us as we move forward in similar prediction tasks. The last point I would like to make, because I'm running out slowly at time, is a really interesting result that we kind of arrived to by analyzing the experimental data. And we're going to call this network effects. The question we would like to ask is that what kind of drug that actually works against the COVID? And the common method to find a drug is a drug that attacks the virus itself, that is binds to the viral protein. And you may have heard this remdesivir, which is under clinical trial and it's a promising drug, is that type of drug. It hits directly the virus. It doesn't do anything with the human cells, but it hits directly the virus. But there are very few viral proteins, so there are a limited number of drugs who can do that. There is the second mechanism, the D2, which is we call it host targeting, but viral host targeting drugs or direct drugs, because it actually hits the direct targets of the virus. So it hits the proteins with which the virus interacts. The promise among those drugs is that it can bind to the human protein in a way that the virus cannot, and therefore will block the virus's ability to infect the cell. What is interesting is that of the 77 drugs that were effective against the virus in the monkey cells, only one drug was of this kind. there's this empty pine. And 77 of the 76 drugs, 76 of the 77 drugs are what we call network drugs, that they don't hit directly the virus, nor do they hit the proteins that the virus hits, but they hit somewhere else in the network. That is, the way they affect the virus's ability to bind is they induce network-based perturbations that effectively alter the virus's ability to enter the cell or to replicate within them. So in a way, the way you can think about it is that if in one neighborhood sits the virus, it hits in the vicinity in a different neighborhood in a way that blocks the virus's ability to attach to the network. And what is interesting is that 76 of the 77 drugs are these network drugs or network effect drugs. And the key is that these drugs cannot be discovered by looking at direct binding to viral proteins, because they don't bind to viral proteins, which is the standard way of how you discover antivirals. Only network-based methodologies can help you discover these type of drugs. So this is probably the most exciting outcome for a network scientist, that these network-based effects are really working when it comes to our ability to block a virus. And really, to discover these drugs, you can only use network-based effects, because direct targeting will never give you that. So I should probably end the talk mentioning that there are quite a number of other ongoing efforts by other labs to use similar methodologies experimentally as well as model-based, including at TU Munich and other places, to kind of look at repurposing opportunities. And if you're interested in this work, then it's our paper is actually available in the archive already, including the new version, just put it a few weeks ago, that has the experimental outcomes as well. And there we talk much more in detail about the methodologies as well. And finally, I will end by acknowledging our collaborators who made this possible. And in the first line, you see the theoretical team, and including Honor, right, who have actually contributed to developing the independent predictive pipelines. And it's no mistake, right, that this is actually the experimental team. And here you see what kind of suit you need to wear in order to do experiments with this virus. And the experiments were done by Robert Dowie and Justin Patterns. And we had two other collaborators, Dina and Joe Loscalzo from Harvard Medical School and Cypher Medicine. For full disclosure, Cypher Medicine is a company that I funded a few years ago. So I have direct interest in that, but they just help with some of the methodologies. Now, I should also end that we have a fabulous team in Boston and now all over the world, but we're always looking for new talent in our lab. So if you have interest in network science, network medicine, AI, machine learning in the network space, we have lots of interest in food and health and science of science. Feel free to contact me or go to our website and we can talk about the possibility of joining us. And this may be a good moment for me to stop and take any questions you may have. Thank you very much for attention.
Thank you very much, László. I'm gonna do the round of virtual applause on behalf of the entire audience, I hope. Talk about, well, we'll take a couple of questions. I'm taking question 0. Like you talk about localization, you showed this artwork that was localized in New York, but it seems to be hanging behind you in Budapest. Is that correct?
Well, that is correct. So that is a work of a friend of mine, an artist from Transylvania that was exhibited in New York. So one of the galleries that I showed you over there has exhibited his work. So I think it was appropriate to show it there.
Very good. Going on to more serious questions, there was one very brief clarification question, namely on the AUC slide. There was circles of different size, and the question was, I think on this slide and potentially on others, what does this mean? That was from Nicholas Landry.
The different sizes were just a visual tool to attract attention to the higher AUCs. And, you know, of course, AUC was included in the axis, but we also wanted to make them a little bit bigger to attract attention to those that have a higher AUC. It's a pure visual trick to induce kind of visibility for those pipelines that work very well.
Very good. Next question from Ishran Kis. Was this analysis for COVID-19 compared to the original SARS? More information might be available for SARS for the validation of different measures.
Absolutely. So obviously the rationale for us to involve in the COVID-19 in kind of March 23 was that there was an imminent need to identify drugs that may help patients. But you could do this analysis for other viruses, not only kind of different SARS type of viruses, but even influenza viruses and so on. In fact, such an analysis has been done by one of my former postdoc, Fei-Shan Chang. And if you watch back this video, the slide that I showed last before the concluding slide, I showed the link to his work where he has done just that. That is that have looked at different type of SARS viruses for which there were already drug targets and validated the network medicine framework as one that could actually allow you to explore drug repurposing opportunities. When he did this work, he was faster than we were because he did that before the experimental results on what human proteins the SARS-CoV attacks were available. So he could only rely on the experimental data that was available for other SARS viruses. We have waited for the moment till the experiments came out and we know exactly what the SARS-CoV attacks, and therefore we could be much more specific for the new virus. We are about to start a project in my lab that we call it the Virome project to repeat many of these methodologies for a whole range of viruses for particular goals. So we'll go back and revisit those questions in the near future.
The next question is from Sergio Lira. Dear Professor Barabási, have you specifically evaluated if your approach could associate COVID-19 with multisystem inflammatory syndrome in children, MISC?
Well, obviously the COVID-19 is very deeply connected to inflammatory diseases because it does, I mean, many diseases induce inflammation, and this has a very, very strong inflammatory component. And we are explicitly exploring that, particularly in the case where we're looking for drug combinations as being effective against the COVID. So this work that I presented has focused only on single drugs, but we believe that drug combinations or cocktails of drugs would be even more effective. And in that case, we are explicitly looking at the inflammasome, which is the subset of the genes that are involved in inflammation, and seeing whether the drug combination could affect that or could also affect the COVID-19 module. We haven't looked at in particular in children. I know that there were these inflammatory disease reports in children, but I think that there's too few evidence for us to go after that to really systematically study. We need to have explicit genes that are over or underexpressed in the case of those patients to be able to do something quantitatively, and I don't think that data is available yet.
Great. The last question is from Daniela Proverbio. I guess for obvious interest, the question is whether hydroxychloroquine has been amongst the tested drugs.
Of course it has been, and I learned a lot about hydroxychloroquine during this process. And one of the things I learned about it is that, and it was confirmed by our own data, hydroxychloroquine is an ethanol because it's very effective to kill in the monkey cells the virus. But the results don't transfer to human cells. What I have not spoken about here, because it's still ongoing, we have another part of the project still ongoing, is that some of the top predictions are being tested in human cells. And we are finding, and others have shown as well, that while hydroxychloroquine is beautifully killing very reproducible fashion the virus in the monkey cells, when it comes to human cells, it's not so effective. And you know, one of the reasons that actually hydroxychloroquine was so highly touted is because every time people did experiments in monkey cells that are faster and easier to do, they found it to be super reliably effective. But because they don't work out in the human cell, that reflects the fact that there are very mixed clinical results when it comes to human outcomes.